Hello everyone and welcome to Instant Biology by Dr. Neelab. Here we are with another very exciting lecture on biochemistry, and this particular lecture would be on the topic amino acids with uncharged polar side chains, and this uncharged polar side chain nature would be established at pH 7. So for those of you who are more comfortable in Hindi, I have already made a lecture on Hindi on the same topic. This particular lecture would be in English, so those of you who understand English well, they can stay with me for this lecture. So today, as I've already said, we would be talking about amino acids with uncharged polar side chains. This lecture would be filled up with diagrams, with figures, with structures, and with easy to understand significant or salient features of all those amino acids that come under this category. So without much ado, let us start. So the first amino acid that comes to mind when we are talking about the uncharged polar side chain amino acids is serine. So let us start with the properties. So when I'm talking about serine, or when for the matter of fact I would be talking about the different amino acids, other amino acids also, I would be talking about the 3-letter codes also, the 1-letter codes also, and also the different salient features apart from the structure as well. So in case of serine, the 3-letter code of the amino acid serine is SER. That means serine is represented by SER. So I have written this, you can check this out. So this is SER, serine. 3-letter code is SER. If I'm talking about the 1-letter code for serine, what is the 1-letter code? It is represented by S. Okay, so 1-letter code is S. Now let us come to the structure of amino acids. Now if you have ever seen the structure of serine, you would identify that it comprises of an OH in the R group. You can see that this is the R group. This is the R group for serine. Those groups that I have highlighted with the yellow color, for example, this is COOH, this is NH2, and this is H. These groups are common amongst all the other amino acids as well. All the other amino acids will have a carboxyl group, they will have an amino group, they will have a hydrogen, and what would be different in this case is the R group. In case of serine, the R group is CH2OH, or you can say that a hydroxyl group is present over here. So this is what I have written over here, that the R group has hydroxyl. What do you mean by hydroxyl? Hydroxyl means OH. That means the R group is comprising of OH. Now if you look at the R group, if I zoom into the R group, this is having CH2OH. You can see this is having CH2OH. Now you would understand this, that OH, the difference between the electronegativities of oxygen and hydrogen is very large. That means oxygen is very highly electronegative, whereas hydrogen is electropositive in nature. This bond is not going to break, but it will be polarized towards the oxygen. That means the electrons would be shifted more towards the oxygen because of the high electronegativity of oxygen. Henceforth, the oxygen will be negatively charged over here, as you can see, and because of the similar reasons, hydrogen will be positively charged, as you can see over here. This is why we call this structure as polar. Okay, so polar means any polar structure would mean that there are two poles. Now you can see that one pole is positive and the other pole is negative. That is why we are calling this structure as polar. Another very important feature when I talk about serine is that since serine is a polar molecule, why it is polar I have already told you. Since serine is a polar molecule, because of that only what will happen is that it will be soluble in water. Why am I saying that it will be soluble in water? The reason behind that is because you see, water is also a polar molecule. Why is water? Why am I saying that water is a polar molecule? If I draw the structure of water, HOH, this is the structure of water, and oxygen is negatively charged over here, partially negative I mean, and hydrogen is partially positive charged over here. Now because of that, again this water molecule is also polar, oxygen being more electronegative. Therefore, both your serine as well as your water molecule is polar in nature. So in biochemistry, we have a rule that like dissolves like. That means anything that is polar will solubilize polar things inside it, and anything that is non-polar would solubilize non-polar things inside it. Since water is polar and serine is also polar, they will be soluble. That serine will be soluble inside water. Now, when something is soluble inside water, or when something shows affinity towards water, then what do you call it? You call it hydrophilic in nature. Hydrophilic. So water soluble, this is water soluble, therefore we are calling it as hydrophilic. Philic means loving. Philic means loving. Hydro means water. That means water loving. So serine, we can call serine as hydrophilic in nature, water loving in nature. Now let us talk about the chirality. Now what do you mean by chirality in case of serine? When I am talking about chirality, for the matter of fact, any carbon molecule would be considered as chiral only when all the 4 valencies that are present on the carbon are different. Okay, so in case of serine, if I check this out, all the valence, if I consider this particular carbon atom, then you can see that this valency comprises of COOH, this valency comprises of an amino group, this valency comprises of hydrogen, and this valency comprises of CH2OH. That means that all of these valencies are different. All of these valencies are different. If all of these valencies are different, then definitely we can say that this is a chiral molecule. And how many chiral centers does it have? If you check this out, does it contain any other carbon that is chiral? No, you would see that only one carbon is chiral over here, that I have already marked. So this is what I have written over here, is that one chiral center is present. Next important thing is that it can get phosphorylated. What do you mean by phosphorylated? Phosphorylated simply means that addition of phosphate group. If I say that something is getting phosphorylated, I simply mean that you can add phosphate group on it. So I will be showing you how a phosphate group gets attached to the serine molecule in just a bit. So the first, the important thing over here is that it can get phosphorylated. Phosphorylated means addition of phosphate. This I have written over here, addition of phosphate. Now, if you have studied about enzymes, then you will realize that which enzyme has the capacity to add a phosphate group. You would understand that serine, suppose kinases basically have the ability to attach phosphate group on any of the compounds. So over here, if I am talking about kinases, more specifically serine kinase, and similar to serine kinases, there are other kinases called as threonine kinases. Serine kinase or threonine kinase, both of these kinases have the ability to attach a phosphate group. Where they will attach? Serine kinase will attach a phosphate group on serine, and threonine kinase will attach a phosphate group on threonine. How? We would be just seeing this. So addition of phosphate I have shown you. This is how addition of phosphate will happen. How it will happen, I am just going to tell you. So all of these properties are related to serine. Let us move forward and talk about how a serine molecule, a serine amino acid gets phosphorylated. So you can see this over here, you would require serine kinase. Apart from serine kinase, you will also require phosphate, PO4 2-. Now you can see this is the OH group, CH2OH. You remember CH2OH was the R group of serine. Now when you use serine kinase and when you use a phosphate group, what happens? This OH is replaced by the phosphate. This process is called as phosphorylation. This process is called as phosphorylation. So serine can be phosphorylated. Threonine can also be phosphorylated. I would be talking more about this in the upcoming sections. So you now understand how can serine get phosphorylated, because it is comprising of an OH group. Now this is the 3D, 3-dimensional structure, basically ball and stick structure of serine molecule. You can see this is the molecular formula for serine, C3H7NO3. Okay, now understand this. This is the general representation, way of representing that carbon is represented by black, hydrogen is represented by white, nitrogen is represented by blue, and oxygen is represented by red colors. So if I show it to you, then this is how the 3D structure can rotate. This is actually the 3D structure of serine. I hope you would have understood this, how this serine is built up. This is the ball and the stick structure of a serine molecule. Now let us move on to the next structure of this particular category. Remember, we are studying uncharged polar amino acids, and in that category we have studied the first amino acid, that is serine. Now the next amino acid is threonine. The next amino acid is threonine. So we will start with the similar parameters that we had studied in case of serine. The 3-letter code over here is THR. The 1-letter code over here is T. Remember, 3-letter code THR, 1-letter code is T. Now let us understand the structural features of this threonine. Remember, we have studied about the serine molecule. In serine also we had seen COOH group, NH2 group, H group, and in the case of the R group we had studied CH2OH. But in this particular case you would not see CH2OH because the R group of different amino acids are different. So what is the R group over here? The R group is this CHOH-CH3. Okay, CH3HOH is the R group basically. This is slightly different than the serine R group, but what you can see, one thing is common, there is an OH group over here also. Now again the same concept applies over here. The R group is OH containing, and since it is OH containing, therefore it is polar in nature. How it is polar, I have just explained it to you while I was explaining or talking about serine. Now since it is polar in nature, therefore again you can say that it is water soluble. If it is water soluble, you can call it as hydrophilic. Next, you can also say that phosphorylation would definitely occur over here. Why? Because OH group is present. So in case of threonine also, just look at this. This is a tyrosine basically structure. In case of threonine also, the OH group is present, and this OH group again can be replaced, or it can be phosphorylated, changed into or reverted into the phosphate group. Therefore what I want to say is that phosphorylation can happen in case of threonine also. This is called as threonine kinase. The enzyme basically that would be responsible for phosphorylation of the threonine is called as threonine kinase. How many chiral centers are present over here? You can see that there are 2 chiral centers. Now therefore this threonine is having very peculiar characteristics, because in most of the amino acids you will see that there is only 1 chiral center. Only 2 amino acids in the entire amino acid family are there that have 2 chiral centers. One is isoleucine and one is threonine. So there are 2 amino acids that have 2 chiral centers, and this is very important. It has been asked in multiple examinations, which 2 amino acids or which of one of the following amino acid is having 2 chiral centers. So answer is isoleucine and threonine. I hope you would have understood this. Okay, 2 chiral centers, phosphorylation enzyme, water soluble nature, OH group, R group, we have understood this about threonine. Now let me show you the actual structure of threonine, actual 3D structure. Let us say that the molecular formula in case of threonine is C4H9NO3. If you check out the molecular formula for serine, yes, it was C3H7NO3. For threonine it is C4H9NO3. So you can see that 1 carbon is less in serine, and also you can see that in case of serine hydrogen is only 7. That means hydrogen 2 hydrogens and 1 carbon is less in case of serine if you are comparing it with threonine. So this is threonine, this is serine. Let us come back to the structure of threonine and find out how does it look like when we rotate it. Check this out. So this is the 3D structure of threonine, again a ball and a stick structure. You can see that and the 2D structure is also visible over here. You can see this is the 2D structure of threonine. Again, black beads or black balls are depicting the carbon structure, hydrogen structure are depicted by the white balls, nitrogen by blue balls, and oxygen by red balls. Let us move on to the next amino acid in this category. The next amino acid is cysteine. In case of cysteine, cysteine is an amino acid that has many peculiar features, and we would be talking about all of these peculiar features in just a bit. So if I am talking about cysteine, cysteine comprises of the 3-letter code that is CYS. The 3-letter code of cysteine is CYS. 1-letter code of cysteine is C. Let us come to the structure of cysteine. The structure is, it has similar properties with respect to the COOH. That means COOH is present just like it is present in other amino acids. Similarly, NH2 group is present just like it is present in another amino acids, and hydrogen is also present just like it is present in other amino acids. So what is the difference? As I have told you multiple times that the difference between different amino acids is the presence of R group. This over here is the R group. So the R group is basically CH2SH. This is your R group. Again, this R group is polar. Remember, we are talking, all the amino acids that we would be talking about today would be polar, but they would be uncharged. Why is it polar? Because there is a electronegativity difference between sulfur that is present over here and hydrogen. Remember, there is a sulfur present over here. So this amino acid is peculiar with respect to the presence of sulfur in its structure. There is one more amino acid that is also having sulfur in its structure, and that is methionine. But in this particular lecture, I would not be talking about methionine for now. Okay, so SH, we have understood this. Now the bond that is present over here between sulfur and hydrogen, it becomes polar. Why? Because of the higher electronegativity of the sulfur atom and electropositivity of the hydrogen atom, the sulfur and hydrogen becomes poles or become poles. Therefore, we can say that the structure of cysteine is polar in nature. If it is polar, it is hydrophilic. If it is hydrophilic, it is water soluble. And one very, very important feature that always comes to mind when we are talking about the cysteine molecule is that it can undergo oxidation. Now, what do you mean by oxidation? First of all, let us understand what oxidation means. So when we are talking about oxidation, oxidation simply means addition of oxygen, or in other words, you can also say that removal of hydrogen can also be considered as oxidation. Removal of hydrogen or addition of oxygen, both are same things. One more important feature over here is that oxidation, it can undergo oxidation, that is fine, but how oxidation can occur, I would be showing you in just a bit. Let us see how oxidation can occur. Now suppose this is one cysteine molecule, one cysteine, and this is the second cysteine molecule. Now both of them are present very close to each other. They are present very close to each other. Now this is one molecule's SH and this is the other molecule's SH. When they come close to each other, oxidation takes place and the hydrogen is removed. I have already told you over here that when we remove hydrogen, this is also called as oxidation. If you are removing hydrogen, then also the process will be called as oxidation. So over here what is happening? Sulfur-sulfur linkage is being formed, and this linkage is called as disulfide bond. Simultaneously, one molecule of hydrogen is also getting removed. So if one molecule of oxygen is getting reduced or removed, then the process will be called as oxidation. So I hope you would have understood this, that in case of cysteine, oxidation will occur. the formation of cysteine will, or when two cysteines will combine, they will result in the oxidation. One more very important thing is that the spelling of cysteine is when only one molecule is present, Cysteine, but when two cysteine molecules they combine together, you write it like Cystine. This is called as cysteine. Okay, so when two molecules of cysteine combined together with the help of disulfide linkage, you will call it cysteine. Okay, now I also said that methionine also comprises of the sulfur group. Does oxidation take place there also? No, the answer is no. Oxidation or disulfide linkage formation will be taking place only in case of cysteine, not in case of methionine. Why? When I would be teaching you the structure of methionine, you will come to know about that. But for now, let us say that in case of methionine, sulfur group is surrounded by two carbon atoms. In case of cysteine, sulfur group was surrounded by or was attached to a hydrogen, right? It was attached to a hydrogen. Therefore, the electronegativity difference between sulfur and hydrogen drove the polarity of the cysteine molecule. But if I am talking about methionine, this is not polar, not very polar. Why am I saying that it is not very polar? Because carbon atoms are present over here and there is not much difference between the electronegativities of carbon and sulfur. Therefore, oxidation is not possible or disulfide linkage is not possible in case of the methionine molecule. This was methionine. However, we have understood this. Now let us talk about the structure of cysteine or the 3D structure of cysteine. Now over here you can see a peculiar yellow colored bead over here. What is this yellow colored bead? It is depicting sulfur. Sulfur is always depicted by yellow color. Now if I want to run this, you can see this is the 3D structure of cysteine, right? And this is the molecular formula for cysteine. This is the two-dimensional structure of cysteine. However, I have shown you the three-dimensional as well as the two-dimensional structure of cysteine. Now let us move forward and talk about the next amino acid, and this is asparagine. So what do we mean by asparagine? What do you understand by asparagine? So asparagine, in case of asparagine, the 3-letter code is ASN and the 1-letter code is N. 3-letter code is ASN, 1-letter code is N. Now let us talk about the structural features of asparagine. It is very easy. COOH is common, NH2 is common, H is common. So what is different? You can see this is different. This is the R group. In case of this R group, you can see that R group is having CONH2 or amide group. You call this as amide group. What is the amide group? It is the CONH2 group. So amide group is present in the R group. This is what we have written over here. What about amide group? Why it is so important or why it is so crucial? Because again NH2 is present. So this is one hydrogen, this is the other hydrogen, and this is nitrogen. You already know that there is marked difference between the electronegativities of nitrogen and hydrogen. Because of that, the hydrogen will be slightly positive in charge and the hydrogen will be slightly positive and the nitrogen will be slightly negative. Okay, I believe you would have understood this. Now let us move forward and discuss about the other features. This is polar in nature. Why? I have told you, because of the positive and the negative poles that are present. It will be hydrophilic in nature. There will be one chiral carbon that will be present over here. How can we say that one chiral carbon is present? This is the chiral carbon. If you check out all the other carbons, you would find that they are non-chiral in nature. Only this carbon that I am showing is chiral in nature. One more important aspect is that asparagine is derived from aspartic acid. Now aspartic acid has its 1-letter code as D. How can I say that this asparagine is derived from the aspartic acid? If you check out the differences between the structure of aspartic acid and asparagine, you would be able to understand the difference. Just let me show you. So yes, just a minute. Yes, so if you check out, this is the structure of aspartic acid. Aspartic acid in the R group has CH2COOH. Let me draw like this. So this is the R group of aspartic acid, R group. So the R group over here is having CH2COOH, but in case of asparagine, this is CH2CONH2. This is the R group of asparagine. That is why we are saying that it is actually derived from the aspartic acid, right? That is why we are saying that it is derived from the aspartic acid, because there is only the difference between COOH and the NH2 group. Okay, I believe you would have understood this. Similarly, there is one more amino acid called as glutamine that we would be talking about. We also say that glutamine has been derived from glutamic acid. Glutamine has been derived from glutamic acid because of similar reasons. Glutamic acid also, you can see that this is having CH2CH2COOH. This is having one CH2 extra if you are comparing it with aspartic acid. But if you are comparing this glutamic acid with glutamine, then you would find that glutamine will have CH2CH2CONH2. This is the structure of glutamine. I hope you would have understood this, right? So I will be talking about glutamine in just a bit. So asparagine we have talked about. Asparagine is derived from aspartic acid. Let me show you the structure of asparagine. This is the three-dimensional structure of asparagine. The molecular formula is C4H8N2O3. Okay, I believe you would have understood this. Now let us talk about glutamine. We have talked more or less about glutamine while discussing about asparagine. There are some things left that let us complete them. So glutamine, one letter code is Q. Always remember the one letter code and the three letter code. Three letter code is GLN. Remember, it is not GLU, it is GLN. Okay, remember this. Next, let me talk about the structural features. NH2, COOH, H, all of these features are common. What is the different feature? It is CH2, CH2, CONH2. Now if you remember the structure of asparagine, you will find it very similar to glutamine, right? The only difference is there are two CH2s present over here. In case of asparagine, there was only one CH2. Now this has been derived from the glutamic acid. Very important point. Why? Glutamic acid was having CH2, CH2, COOH, and glutamine is having CH2, CH2, CONH2. This is the main difference. Now let me talk about the other properties. Since NH2 is present, again hydrogen is slightly positive, nitrogen slightly negative, this again is slightly positive. This will be called as hydrophilic amino acid. Polar in nature. One chiral carbon is present over here. If you check out the other carbons, they would not be chiral. Only this particular carbon is chiral. One chiral carbon. And I have also told you that in the entire amino acid family, there are only two amino acids that have two chiral carbons. One is isoleucine, one is threonine. Okay, now this we have understood. I also told you about the similarity with the glutamic acid, so deriving it from the glutamic acid. This is the structure of glutamine. You can check this out. If I run the 3D structure, this is the 3D structure. You can assume the structures, and you can also see that this is the molecular formula for glutamine. This is the 2D structure of glutamine. Let us move on to the tyrosine, last structure of this particular class. We are talking about tyrosine over here. The three-letter code of tyrosine is TYR. The one-letter code for tyrosine is Y. Remember, again I am saying again and again, there are some amino acids whose one-letter code or three-letter code you might not be able to identify by the original name. For example, arginine. Arginine does not have the three-letter code as ARG. Okay, it does not have, sorry, the one-letter code as A. because A has been given to alanine. The 1-letter code for arginine is R. Similarly, for other amino acids, for example, aspartic acid. Aspartic acid does not have the 1-letter code as A. It has 1-letter code as D. Okay. So these are important things. Now, if I'm talking about tyrosine, 1-letter code is Y. Again, it is not being depicted by T because T has been given to threonine. It is depicted by Y. So 3-letter code is TYR. Let us talk about the structural features. This is COOH, NH2H. These are the common features. What is left? This is the R group. Now you can see that this is having CH2, benzene ring and an OH. Therefore, we can also call it, this is a very important point, you can call it phenylalanine. Why can you call it phenol? Why are you able to call it phenol? Because phenol, this is phenolic groups, right? Benzene with OH, and this is the CH2 group. You already know, I have already talked about the non-polar amino acids, and in the non-polar amino acids we had studied the structure of alanine. Alanine has an R group of CH2. Therefore, we are clubbing both over here. CH2 group is present, therefore we can say alanine properties. This is the phenol group, therefore we can say phenolic property. Therefore, we can call this as phenylalanine. The benzene ring is present over here, you can see this. Again, you can see this is the OH group. If OH group is present, this is polar in nature. Oxygen slightly negative, hydrogen slightly positive. Since it is polar in nature, you can also call it hydrophilic. One chiral carbon only will be present, but since this is having the benzene ring, I have told you multiple times that whenever a benzene ring will be present, it would be showing absorbance. Why? Because of delocalization of electrons. Suppose this is the benzene ring, and what happens is, when a photon light falls on it, what happens is it will definitely absorb this light. Why? Because with the help of the energy of this light, what will happen? It will change the conformation. How will it change the conformation? Let me show it to you like this. So the bonds will be changed like this. This would be the new conformation of the bonds, right? I believe you would have understood this. Therefore, we always draw benzene like this. So the energy that was absorbed was used for the delocalization of electron. Let me write it down. Delocalization of electrons. Electrons have been delocalized. So at what wavelength tyrosine absorbs? 274 nanometer. What is the absorbance that is obtained? It is 1.4. These are very, very important typical features of tyrosine. Now let us check out the structure, 3D structure of tyrosine. This is how tyrosine would look like. There is a benzene ring. Clearly you can see over here, this is the benzene ring, right? So this is the structure of tyrosine. The formula is C9H11NO3. And with this 3D structure, we come to the end of all the amino acids that were present in this particular family, that means polar uncharged family. I hope you would have loved this lecture, you would have liked, and you would have learned the structures during the lecture only. You would have understood the salient features, the 3-letter codes, the 1-letter codes, and all your confusions would have definitely resolved with this lecture. If you liked the lecture, please hit the like button. Do not forget to subscribe to our channel. Please share this video with your friends as well, and do not forget to hit the bell icon so that you can get all the updates instantaneously. Thank you so much. Have a good day.